So we're going to walk through chapter 31 in this video. We begin here at the cover page where we see an image of the ribosome. It's a really huge molecular assembly of RNA and proteins, and you can divide the structure into two parts: the 30S subunit, which is responsible for decoding the messenger RNA, and then the catalytic subunit, which is the 50S particle, and it's responsible for catalyzing the chemistry of peptide bond formation. And you see here the polypeptide. Each little bead represents one amino acid residue in this polymer of this polypeptide. And the chemistry, the substrates are these aminoacyl tRNAs. Here's number one, and a second one comes in, and after the chemistry has been catalyzed, this acyl group on tRNA number one gets moved onto the amino group of the second tRNA. So we're moving this amino acid onto the amino terminus of this aminoacyl tRNA, and that process then continues. A third aminoacyl tRNA comes in, and we move now this growing polypeptide. The acyl group moves onto the amino group to form a new peptide bond. So here's the peptide bond made in this arrow, and then here is the peptide bond made in this first arrow. And the table 31.1 points out that we need to be very accurate with each step of protein synthesis. We can tolerate a certain frequency of inserting the incorrect amino acid, but because we're doing this chemistry so many times, that gets even a fairly accurate enzyme like that only makes one mistake for every 1,000 incorporations. For a polypeptide of 100 amino acids, we're only going to be only 90% of those molecules will be error-free. And if you have a really large protein, the chances of getting an error-free protein are quite low. So a tolerance of about one error in 10,000 leads to fairly accurate proteins, even for these large-sized proteins. And the job of making sure that things are accurate has to do with relies on the structures of tRNAs, and there are several representations of these tRNAs. Figure 31.3 and emphasizes the stem loop structures, and the 3' hydroxyl group. This is where chemistry is happening. The amino group of the amino acid will get attached here. Here's the anticodon loop. These side images illustrate how many of these bases, especially in the loops, they are modified to be different from the standard Watson-Crick type bases. And over here is figure 31.5 emphasizes the three-dimensional form, and also 31.4 emphasizes how they form these L-shaped molecules. The anticodon loop is quite far away from the place where chemistry is happening. Finally, we're going to end this first portion looking at what's called the Wobble hypothesis at the third codon position. So here's a codon 1, 2, 3 positions in the messenger RNA, and that is being interpreted by the bases in the anticodon, and this is an anti-parallel RNA to RNA base pairing. The first two codon positions are interpreted very precisely. You have to have A paired with U and C paired with G at these first two positions. But at the third position, the ribosome tolerates mispairings or alternative pairings. And the table 31.2 tells you what each of these first anticodon positions is capable of decoding. So if you have C, according to the Wobble hypothesis, if you have C at this position, Z prime, then Z must be G. So that's a strict rule. And if you have A at this first anticodon position, then you have to have U. If you have U at this position anticodon, now you can interpret both A or G. And the G at this position can interpret both U or C at the third codon position. And then inosine is illustrated here in red. It looks a little bit like guanine, but it's missing its exocyclic amine group, and it is generated by the deamination of adenine. So inosine is able to interpret 3 bases in the codon. So if inosine is here at the first anticodon position, then you can interpret U, C, or A. And this is just a hypothesis. Our codon table with dots summarizes data from experiments showing that the Wobble hypothesis can explain some of the patterns, but not all of the patterns, and that uracil often can interpret more than just these two bases. Over here, 31.7 explains why base pairing is so strictly enforced at these first 2 codon positions. It has to do with steric complementarity. The bases are being examined in the minor groove by a component of the 16S ribosomal RNA. So only precise Watson-Crick type, here in this case adenine base pairing with uracil, and the anticodon position, the codon. So this belongs to the tRNA, this belongs to the messenger RNA, and this nucleotide here belongs to the ribosomal 30S particle, the part of the ribosome that is responsible for decoding codons.